Biomedical AI speaker series. Today we have Kenza Bouzid. She's a senior Microsoft researcher working at the biomedical imaging team at Microsoft Health Futures. Kenza also has a master's from KTH in machine learning. And today she's with us to talk about her recent paper looking at insights into a radiology-specialized multimodal large language models with sparse autoencoders, specifically Myra 2. So thank you so much, Kenza, for joining us, and we're excited for the talk.
Thank you. Thank you for the invitation. Just wanted to thank the Harvard Medical AI Club for the invitation, and I hope this talk will be interesting for everyone attending. Hello everyone, and thank you for joining us. I'm Kenza Bouzid, as Mohammed mentioned, a senior applied researcher at Microsoft Research Health Futures. I'm based in the Cambridge, UK lab. It's a great pleasure to be talking to you today about my work on insights into a radiology-specialized multimodal model with sparse autoencoders. Before we go on, I just wanted to give a little shout out to the Health Futures UK team. These are the people behind this work and behind all of our efforts related to radiology reporting and our Myra efforts in general. You can find more details following this link about our ongoing work and also about this work. Okay, let us start with some background and related work. Signals for interpretability have been around for a long time, but since May 2023, we've seen some early signals for that could enable large-scale automated interpretability. And for instance, OpenAI has demonstrated that LLMs could be explained, or at least LLM neurons could be explained by LLMs themselves. This really paved the way for large-scale automated interpretability, and as we can see over here, they show an example where they give GPT, they highlight the tokens that activate a neuron and ask it to come up with an explanation for this specific neuron. And we can see here it's related to words and phrases related to performing actions correctly or properly. Later on in May 2024, Anthropic applied sparse autoencoders to entangle neuron activations in Claude 3 into an abstract but still human interpretable concepts. This was all based on a few hypotheses that a single neuron or single neurons in LLMs in general are polysemantic and can encode multiple features that are composed linearly in the feature space. So what they show here is that they've been able to extract millions of features from Claude 3 Sonnet. They were able to interpret them and find that they were generally interpretable and monosemantic. And they also showed that you can actually use these feature explanations into multiple downstream tasks, such as classification and steering model behavior. But I do want to highlight that actually SAEs have a lot of limitations. And also the underlying hypotheses have their own limitations, and it has been extensively studied in the literature, and a lot of questions have been scrutinized. So are these features always one-dimensional? Some people looked into whether SAEs are the optimal way for model control. And we've seen multiple work addressing the fact that we've seen some feature splitting and absorption in SAEs. In addition to multiple works related to the architecture itself and how we can improve it. And since then, I think SAEs have been applied to various domains and models, like GPT-4, Gemma-2, Llama-3, RAD-Dino, which is a radiology model that our team has built, Pluto, which is a pathology model, MammoClip, mammography model, and so on. So we thought about perhaps applying it to our own model, MyRA2. As we built this model that is purposed for grounded radiology reporting, we had a grown interest and curiosity to actually better understand the inner workings of this model. This is a lava style model, so a vision encoder that gives us a visual representation of the images that then get projected and aligned to the language space through a linear layer. And then you train it for various tasks like findings generation or grounded report generation. In this context, we pass in the three different images: the prior report, the lateral view, but also the current image, and also some additional metadata like the indication, the prior report, and so on. So the question that we try to tackle in this work is whether we can use sparse autoencoders to build a better understanding of MyRA2. Which led to this paper that we've presented at the Actionable Interpretability Workshop at ICML in Vancouver this year. I just put some links here for the model on Hugging Face and the archives so that you can read more details about this work. All right, so before actually we look into this, I wanted to spend some time explaining what is a sparse autoencoder for those of you who are not familiar with this work. It's built on two main hypotheses. The first one is that neurons are actually polysemantic. The idea is that each individual neuron may represent a mixture of unrelated features, and that is because the data contains a lot more information than the number of neurons available in the model, which leads to some features being superposed in a single neuron. And the linear representation hypothesis comes up and says that these different concepts that are superposed within a single neuron are actually composed linearly. And there is a linear structure in the activation space that could be easily decomposed. So the idea then is to use SAEs to decompose this superposition into a sparser and larger latent space that is promised to be more interpretable. And yeah, like the polysemantic nature of neurons makes them very hard to interpret. So the idea is to split them into smaller directions so that we can easily interpret them. The decoder over here is then thought of as if it encodes different concept directions, and they are all linear. And it can be trained for reconstruction depending on the architecture that is used. You can use a sparsity penalty or a constraint, like for example in top K. And only a small number of these latent feature will be active at once. Okay, so for MyRA2 specifically, what we do is we feed in the training data examples from MyRA2 that are extracted at the residual stream. I'll go into more details next. And then we use a Matryoshka SAE to decompose this residual stream. For the data preparation, we look at the layer 15, which is the middle point in this model. This is a Vicuna, I think, 7 billion model. And we picked the middle layer because there's some prior work showing that the middle layer is the one that encodes more abstract and interesting features. And what we do then is extract activations at all tokens, but then we apply some filtering to just like remove all boilerplate tokens like the system message and the start and end of sequence and so on, which are not that interesting to look into. This gives us 34.7 million activations, and this is what we use for training the SAE. I mentioned before we use a Matryoshka SAE. The idea is that we have nested autoencoders at different levels, and we train them simultaneously to reconstruct the representations of Mara2. As you can see here over from the shades, each level is trained to, I'm not sure if you can see, oh, actually, I'm not sure if you can see my pointer. Each level in the nested autoencoders will reconstruct the full representation. So the objective is enforced at multiple levels at the same time. It is thought to be slightly more interpretable, and it was the current SOTA at that time. I'm sure there has been a lot of advances since, and it worked reasonably well. This is like a Matryoshka batch-top K, and the K, we've set the K to be 256. The choice was mainly because we have it's a multimodal model. It has images and text, and they all contain a lot of concepts, and that's why I think our case probably on the higher side compared to previous work, but most of the previous work actually was applied in language, and it's usually set to 32, but that's because like in language you won't have more than 32 concepts at a time. For all of the results that I'll be showing, we used expansion factor 4, which resulted in 16,384 possible interpretable concepts. All right, now that we have these SAE features, how can we interpret them? We use techniques from automated interpretability, leveraging LLMs to come up with an explanation for each feature. The way we do it is by taking, for each SAE feature, we identify different samples that activated with different activation strengths. So that's the little weights that you see over here. And we present them to an LLM, in this case GPT-4o, and ask it to come up with an explanation that justifies the fact that this activation is higher for some samples and lower for others. So we also pass in samples with 0 activation so that the LLM has some contrastive samples to compare to. So you can see over here the prompt that is used to come up with these explanations. And really what we care about is a concise explanation for the pattern identified in the different samples, and also a rationale just to make sure that it's justified well enough. Okay, now that we have these explanations, how do we actually make sure that they are correct and that they are more or less of good quality? To have some type of quantification for that, we use detection scoring, again, using LLMs. The idea is to reverse the task. Now that we have the explanation, we ask the LLM to come up with the activation. So given, for example, over here, positioning or change of lines and tubes, we ask the LLM to detect whether this specific feature or concept would be active for the sequence that we show it. And to simplify the task, we just turn it into a binary task. So we don't look for the exact activation strength, but only whether it was active or not, as a measure for the quality of the interpretations. I do need to mention, I think I've skipped that. We actually did not use images for the auto-interp, and I think that's one of the main limitations of our work. So all we showed it is the text, and it could probably be improved through the use of images. All right. Oops, I skipped a lot of things. Now that we are, we were talking about detection scoring. This is the prompt for it. All we care about is return 1 if the example demonstrates the property, and 0 otherwise. From there, we can run this analysis on the full 16,384 features and get a measure of precision, recall, and overall F1 for all the features, the explanations. And surprisingly, we found that a very small number of these features are actually highly interpretable, so above 75 F1. Only 288 features were actually very highly interpretable, and most of the rest was, mixed feeling explanations. When we first found this out, we were quite surprised. We looked into prior work to try to compare more or less how good it is in the literature. And it wasn't very clear because most of the prior work didn't characterize all of the features. So they didn't apply systematic quantitative evaluation for all of the features and only cherry-picked a few. Whereas we tried to do it at scale for all the 16,000 features. I'm showing over here a few examples of these features. So the feature 1599 is responsible for describing findings without comparison to prior images. This is actually a very interesting one. Because we see a lot in radiology reporting what we call temporal hallucinations. So models tend to generate comparisons even if like there is no prior report. So actually this one was quite interesting to find so that we can control for that actually. We also found some other features that are related to pathologies like pleural effusion, for example. The feature 6412 related to detection of pleural effusion on imaging studies, and some other ones. 3716, for example, is also quite interesting. Presence of shortness of breath as an indication. Since we pass in the indication as well, we can also control for that. What we also did is look into feature statistic. This is mostly to sanity check the feature density and the explanations. The feature density is defined as the fraction of samples on which a given feature activates. What I'm showing here is on a random subset from the training set, so just 500,000 data points. Remember, a data point in this case is an activation at a token level. And what we've observed is that a very small numbers, maybe let me try to point to it over here, a very small number of features activate on almost all the samples. And we also found that these are commonly occurring features. And they're actually not very well explained. I think we've got a few examples in the paper, more concretely, the explanations for those specific features. And we haven't observed any correlations between feature density and interpretability. All right. Now that we found SAE features and we've been able to label them, what can we do with them? One downstream task that we've considered is steering. Steering is, you can think of it as model controlling. So you can steer the model toward a direction. And since these are linear directions, we can very easily manipulate the internal representations of the LLM to emphasize certain concepts. So the example I'm giving here is related, I think, lines and tubes probably. And we want to push the model to generate things that are related to lines and tubes. And the way we do it is by taking the concept vector of lines and tubes. It's actually quite simple because the SAE decoder is just encoding concept directions. So once you know what a feature corresponds to, so when we identify the lines and tube feature, we can simply take the concept decoder rate that is corresponding to that feature and add it to the hidden dimensions of the LLM in the residual stream. We do that at each token generation, so for each next token generation, and all along the sequence. And to validate that, again, we use LLMs to identify whether the steering worked and resulted into on-target effects or off-target ones. So yeah, as I've mentioned earlier, the feature is related to positioning of change or lines and tubes, and we want the LLM to judge whether the output is modified such that this specific feature is emphasized and whether there was no other unrelated changes. So we give it the steered output and the original output and get it to judge whether it resulted in on-target or off-target effects. Perhaps a few examples of this. Feature 1599 that I've described earlier related to describing findings without comparison to prior images. So from the original generation, you can see over here, the report do contain a few comparison mentions. So compared with the prior study, we can see here that the lung volumes are lower and the cardiomediastinal and hilar silhouette are unchanged. By steering the model to emphasize this feature, we actually have a steer generation that doesn't contain any comparison mentions. And in this case, we only had on-target effects and no off-target effects. In the second case with feature 6412, that is related to pleural effusions on imaging studies, we can see that the model correctly added a left pleural effusion. So the original generation didn't have any pleural effusion, so it said no pleural effusion. And the steer generation adds in a small left pleural effusion. Now, the interesting thing is that, well, interesting, arguably interesting, we see that it also added an atelectasis. This is actually an interesting one because there is a known correlation between pleural effusion and atelectasis. And when we steer the model to generate towards the feature for pleural effusion, it also added an atelectasis. This is probably related to the underlying distribution of the data. So whenever pleural effusion is present, there's also an atelectasis. So it is an off-target effect, but perhaps a natural one. We do this systematically on all of the MIMIC validation sets, so about 7,000 studies. And we show here a plot. I'm not sure why I blurred this. We can see here only on-target effects, both on-target and off-target changes in blue, and in yellow, only off-target changes. I think the surprising thing for us, again, is that it didn't work for all the cases, but there is an argument for it to actually not work in all cases. Let's take the example of pleural effusion, for example. If there is already a pleural effusion, what does it mean to steer the model to generate more pleural effusion? Similarly, for other features related to cardiomegaly or so, I think all what I'm trying to convey here is that you need to be very careful with the sample selection before you apply steering, because steering doesn't always make sense in all cases. And that's why it doesn't work if you just apply it systematically for all samples. And I think this is quite consistent with prior work. It doesn't always work, but it's actually quite interesting because it could be applied to, it could be used in like a hospital, for example. Let's say if we know that the model is not great at certain pathologies, we can try to make sure that the model won't generate any of those. Just to avoid confusion. And similarly, if we want to adapt it to another hospital style, we can manipulate the features that are related to radiology style and so on to make sure that it's more in line with the style of that specific hospital. In summary, what we did as a study, we extracted activations from IR2, trained a sparse autoencoder, interpreted the SAE features, and steered it, the normal autointerp pipeline. We found that we can find some interpretable concepts in IR2, but not many of them are actually interpretable. And I think there are, some arguments to be made that maybe using the images could help make that better. We've shown that we can steer mirror to generation, but not always, which is actually valid in some cases. And we learned that interpretation is actually probably the bottleneck of this whole pipeline. We need perhaps more sophisticated methods for the auto-interp to actually find the right explanations. SAEs in this case worked, but also have limitations in concept discovery, so you can imagine other methods as well. Steering and scenarios when it actually work are not well characterized. So all what I'm trying to say is that there's still a lot to do, and it's a very interesting research direction to pursue to better understand these models. Mirror to SAE is available on Hugging Face. We've shared the SAE weights for 3 different expansion factors, 2, 4, and 8, and we also share all of the auto-interp results, so you should be able to find the explanations for all of the features for a specific expansion factor. Thank you all for listening. Put in again the few links in case you would like to look into the details, and I'll open the floor for questions if you have any.
Hi. Maybe I can ask a question if no one else wants to go. Thank you so much for the talk. I found it super interesting. So one question I have, more of like a general, broader question, maybe in general about SAEs. Do you feel like the insights that you got from looking at these SAE concepts, I mean, you can use them for steering. Do you think, or do you see any pathway how you could also use them maybe to improve pre-training or post-training altogether to get a better language model in the first place? I mean, of course, this would be more effort, but do you see any pathway for doing this, or SAE is not good enough for that yet?
Yeah, I think that's actually a great question. I think there's an argument made for that, and there's been some work. I believe Anthropic looked into that with cross-coders so that you can monitor the trainees and also at different layers. It is very expensive, obviously, but if you would like to get a very good language model, it could be a proxy to get some insights on what features are learned at different, even like for training monitoring. You can check like different checkpoints during the training and find the optimal one that has like learned the most concepts that you care about. You can also look at the training dynamic, how these concepts change during the training. Yes, I feel like there are a lot of downstream tasks beyond steering for SAEs. You can also use them for circuits discovery to get like a better understanding. So far, we looked into just the concepts, but you can also try to understand how these concepts are used to come up with the generations. But then it's a bit expensive because you probably need to do it at different layers. And it's a lot of, tracing all of the mechanisms in the LLM could be quite challenging, but I think it's actually a very interesting and challenging research question in the mechanistic interpretability world that is all about reverse engineering models. So yeah, I think beyond steering, there are a lot of other downstream tasks.
Thank you so much.
You're welcome. I guess if there are no other questions, we can also resume here.
Hello? Can you guys hear me?
Yes.
Okay. Sorry. Okay. I think we have time for some questions. So if anybody has questions, they can ask.
Hey, do you hear me?
Yes.
Excellent. Thank you so much, Kenza, for your presentation. Nice to see that and to hear that. And I'm wondering if you could give us like some details about the data itself, the size, type, if we can, for example, apply the same methods for MRI data.
Yeah. Yeah, just to give you an idea on scale. I was trying to find a slide, but completely failed. So what we have here is, what we've done is we've used all of, I'm not sure if you're familiar with MIMIC data, but this is 150,000 samples. So like radiology report studies. And then the 35 million is coming from the fact that we extract activations at each token. So the sequence length is around 4,000 or so. And you just like time it by the number of samples, and that's the type of scale that we're dealing with. We also applied some filtering to remove any boilerplates, tokens, and so on. But I think, yeah, this is like the scale we were dealing with. For MRI specifically, are you thinking about the image encoder probably, right?
Yeah.
Yeah, I've seen a lot of work on that as well. In fact, I think there's been some work on our own vision encoder by Dino. I'm trying to find the slide. Over here is the reference here. So you can apply it very similarly. There's been a lot of work on applying SAEs for visioning models. And I believe the bottleneck really is the auto-interp. Once you have the SAE features, since you can't really rely, I'm not sure how GPT does on MRIs, but it might be a bit hard to auto-interpret them. You can rely on, if you have some paired data, for example, reports or any metadata or like segmentations or so, you can use that as a proxy. But it could be great to gain some more insights into your model.
Okay, and how many hours did this take you to train the
This
model?
was actually quite fast. On a single A100, I think a couple of hours, maybe take 4 to 8 hours, depending on the expansion factor. Our scale is probably a lot smaller than what other prior work does, especially in the text domain, because the training corpus is much larger. But in this case, it was quite fast to train.
Excellent. Excellent. Thank you so much.
You're welcome.
Any other questions? Okay, one question I have is so this is my like third or second ever interpretability paper I've ever read. So because I don't really usually read this, and I think I already read it because it was directly related to radiology and the task that I'm working on, which is grounded report generation. But I'm working on it for CT. But have you looked at other techniques for getting interpretable features like concept bottleneck models? I know concept bottleneck models. I know I think you need ground truth for that. But I think there's a paper that I read from Stanford a couple of years ago that they can do unsupervised learning for generating concepts, and there's like a plethora of other methods. So have you
Yeah, definitely something we're very interested in our group. We're exploring. There's still some work in progress that I cannot talk more about, but it is a very interesting direction, because once you have these concepts, you can also use them for building an inherently interpretable model, like a concept bottleneck model. I think in this case specifically, it might be a bit hard because it's a generative model. We're training it for report generation. But if the downstream task is like a classification, then you can very easily build a CBM type of model to do that. I think there's also some work on turning LLMs into like concept model. There's like some work from Meta, large concept models. But I quite like the direction of anti-hoc explainability and just baking the interpretability into the models to make them slightly more transparent. And yeah, like the combination of SAEs and CBMs have been very popular in the last few months. There's been a tremendous amount of work going into that effort. And I think I was, in fact, just reading a paper about that for histopathology specifically, but it's been applied in various domains.
Okay, and is there because I've heard people say that there's a trade-off between performance and interpretability when you try to bake it into the model. Is that?
Yeah, it is. That's actually a good point. You might lose into some like performance, but recent work has shown like at least competitive results with just completely black box models. So I think if you enforce like the interpretability through a concept bottleneck model, you just like lose some of the things that the model might use to shortcut some correlations, and just yeah, there's a trade-off obviously, but it really depends on what you care about. Very high performance, or you'd rather have a model with like slightly lower performance but better interpretability, which I believe most healthcare applications care a lot about.
And is that because, so I think intuitively the reason why models lose performance when you make them interpretable is because there's some parts of the model where it's relying on signal that we don't usually consider to be used in this task, right? But is that because this signal is just noise, not noise. I don't know what's the correct word. Like something inherent in the dataset, but it's not generalizable. Forget what the correct word would be.
Yeah, I think really what happens is, at least with CBMs, is that you then enforce the model to only make the prediction from the concept layer. And these concepts are predefined. So if you haven't thought of something that is not directly known in this concept layer, then the model might not be able to make that link. And you just lose that signal that otherwise is hidden in the black box machine, if that makes any sense.
Yep, that makes perfect sense. Okay. If there's no other questions.
Yeah, please, another quick question.
Yeah.
Like, Kenza, as a team there in Microsoft in Cambridge, UK, how much are you open to collaborate, for example, here with Boston Children's Hospital?
Very open.
Okay.
We encourage, I'm not sure what's your background or stage at the moment, but we do have, we take on interns, and we have openings for jobs, but also in general, we try to collaborate with academia. So it doesn't have to be through a job or so. We can also just talk, and if we have similar interests and like projects that could be interesting to work on together, we're very happy to collaborate.
Okay, excellent. So I'm about to email you, and if it is possible, we can talk off to discuss this, please.
Yeah, please reach out. I think I've put up my email Someone?
Yeah, I got it. Yeah,
Yeah,
I got it. Yeah.
perfect.
Thank you. Yeah, thank you so much. Thank you so much.
Welcome.
Okay, we can end here, I guess. Thanks everybody for joining us. Oh, one last question. I know in Cambridge, UK, I know Ibrahim Hamamshi was there last summer,
Oh, yeah.
right?
Yeah.
Were you guys in the same team, or was he?
Yeah, he was in my team. We weren't working on the same project, but he was in my team.
Okay, nice. You guys know each other?
Yeah.
Okay, perfect. All right. Thank you so much, Kenza, for joining us, and thank you
Thank you.
everybody for attending. This talk and other HMAI talks are going to be on YouTube, so you can rewatch it whenever. And yeah, thanks everybody, and see you guys in the next episode. And thank you, Kenza, again for
Thank
joining.
you.